You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast, and I have Dana Carroll, PhD. He's a distinguished professor in the Department of Biochemistry at University of Utah School of Medicine, and a member of the Nuclear Control of Cell Growth and Differentiation program at the Huntsman Cancer Institute. So, Dana, thanks for coming. How are you doing today?
I'm doing fine. Thanks for having me.
Yeah. Well, I had seen some publications about some of your work. It was dealing with using various mechanisms to, to repair genetic damage. A term mentioned was zinc-finger nucleases and CRISPR and Cas9. So maybe you can let listeners know what's your work about, and then we'll get into the details of how it works.
Sure. The work that my group's been doing, and many other people around the world now, is working to be able to change individual genes in the DNA of essentially any organism. And the tools have developed over the last 15 or 20 years. The zinc-finger nucleases that you mentioned were the first of the tools that allowed us to get access to specific genes in the total genomic DNA of individual organisms or cells. And the current tool that people may have heard about is called CRISPR, or CRISPR-Cas, and it has made things a lot easier. And by now there are hundreds of different animals and plants that have had their DNA modified using these particular tools. A lot of what's been going on has been just for research to see how particular genes work, and what controls the development of individual organisms. But in the last few years, there have been more and more applications that will have an impact on the public.
So I have a question. A gene, I've heard from some people that it's hard to define a gene. I mean, it's really more accurate maybe to say a series of nucleotide bases. And the reason I've heard this is that, I don't know, if you have a sequence of, let's say, 1,000 nucleotides, I had thought that there's overlaps, like if you label them 1 to 1,000, that's one quote unquote gene, but it can also be utilized as 500 to 1,500, let's say. And part of that sequence is utilized and part of another sequence is utilized, and that constitutes quote unquote another gene. Is that the case, or am I wrong about that?
It is fairly complicated. I don't know whether all of your listeners will be familiar with the fact that in humans and in many other animals and plants, genes as we see them in the genome, in the total DNA, are made up of what we call exons and introns. And only the exons actually are coding sequences for proteins, and the introns are sequences that interrupt those coding sequences. And we don't really know what their function is in most cases, but corresponding to what you said, sometimes from one particular gene, different exons can be used, spliced together to make a messenger RNA that encodes one protein or a different messenger RNA that encodes a different protein. So a single gene can be used to make different but related proteins. So that's one complication. Also, the sequences of a gene that will ultimately be used to make a protein have to be regulated somehow. The gene needs to know when to be on and when to be off. And so there are DNA sequences that regulate the expression of a gene. So are they part of the gene or not part of the gene? And sometimes they can be quite complicated and fairly far away from the gene in chromosomal DNA. And then there are some genes that we have a hard time really identifying because they look a little different from normal. So defining a gene, just as you said, can be fairly complicated.
I guess there's also then epigenetics, methylation and histone deacetylation that affects how the gene is expressed as well.
Absolutely. That provides another complication. But, the more people study and learn about genes and their expression, these things are beginning to become clear. We really didn't have much of an idea about many of the epigenetic controls 15 or 20 years ago, but now not only do we understand more of those, but we're able to actually manipulate them the way we manipulate DNA sequences. Again, people are using the CRISPR technology to do some of this.
I could see if you sequenced someone and you saw what was, phenotypically normal, and then they were abnormal in a certain sequence, and you use CRISPR-Cas9 to go cut that sequence out and put in the quote-unquote normal sequence. There's probably little fear of issue. But how do you know if you're cutting out a sequence that it won't have all these side effects, these ancillary effects that you had no clue about if you're doing a knockout or something?
Yeah, that's a concern. We aren't so good right now at making corrections in the way you just said, to remove what looks like a bad sequence and put in what we think is a correct one. We're not so good at that right now. So that's one of the areas of research where people are putting in a lot of effort to try to make it possible for us to efficiently make corrections of that sort. But even when we are able to make corrections, there are worries about not just doing the correction, but doing some things that would compromise that person's genome. So that's something that we're working on. And I have to say that the extent to which you care about what we call off-target effects really depends on what you're doing. So for some really devastating diseases where there may not be an alternative treatment, we might be willing to risk some off-target problems to get a good correction of the target that we're aiming at. So, people are concerned about off-target effects, but the extent to which you are concerned depends on what you're doing and what you might be concerned about.
So why are corrections so hard? Is it because that corrections will unintentionally act systemically, or is it because they're hard to get them to act anything but locally? Like, what are some of the complications?
When we use CRISPR or the zinc-finger nucleases, what enzyme does is it makes a break in DNA, and we're able to direct exactly where that break will be. Once the break is made, everything that happens after that depends on DNA repair activities in the cell that's being targeted. And we don't have complete control over those repair activities. Some cells are pretty good at the correction style of repair, but many other cells aren't very good at it. That is, they have other ways to correct or to repair the break that may lead to some mutations right at the break. We can make use of those in some circumstances, but to make the correction, we're dependent on what people call homologous repair or homology-dependent repair. And as I said, there are a lot of cells out there in different organisms that just don't have that capability really cranked up to be efficient. So one of the areas of intensive research now is to look into what do we have to do to a cell so that it's doing the homology-dependent repair much more efficiently, so that it will do the corrections.
Well, what if I have a condition where, like, my liver can't process something for some reason? So you want to repair my liver cells so that they can. If you do such a thing, will let's say you're injecting material that does this, CRISPR-Cas9. What will happen to the other cells in my body? Will it naturally go and cut their DNA sequences, or kind of stop locally? And what are the effects if so?
That's a good question. It's another area where a good deal of research is being focused. A lot of what we're able to do now is much more easily done outside the body, and that's called ex vivo therapy. I'll give you one example, then we can come back to the delivery issue that you were just talking about. With one of the diseases that people are working hard to try to provide a therapy for is sickle cell disease. This is a disease that affects red blood cells in the circulation. It's quite prevalent in many parts of the world, and in areas where there aren't good symptomatic treatments, children who have this disease tend to die quite young. So the idea, which is just being acted upon, not in the developing world, but in the developed world right now, people are taking cells from the bone marrow of sickle cell patients, doing a correction or another treatment that would reverse the defect in red blood cells, doing this in the laboratory, and then putting the cells back into the patient from which they came. So it's essentially an autologous bone marrow transplant. You get bone marrow cells from a patient, you provide a therapeutic correction, you put them back into the same patient. And that's more easy to do than treating cells in the liver of a patient who has liver disease, or in the bone marrow of a patient with sickle cell disease. We don't have wonderful ways of getting efficiently into individual organs. So one worry is that the efficiency is not so good when we're doing this in vivo in a patient who has the disease. And another worry is that we may be going to other organs, as you suggest. The liver is actually a pretty good target because lots of things go to the liver and not so efficiently to other organs. There are ways to try to direct the therapy to a specific organ. One way is to try to do an injection of the therapeutic materials. That's being done in muscle and also in the eye. And so if you can do an injection into an individual organ, you worry less about the whatever the materials are going to other organs in the body.
Well, even in ex vivo applications, how many cells let's say you're treating the bone marrow or sickle cell anemia, one cell I'm sure is not enough, but how many are needed? What percentage of cells? And, can you do too many or too few? And, what's been explored there?
Well, we can get a lot of cells from bone marrow. I want to say hundreds of millions, tens to hundreds of millions of cells. These are stem cells that will go back into the bone marrow and reconstitute the ability to make both red and white blood cells. In the laboratory, in these ex vivo treatments, there are procedures now that are effective in making therapeutic modifications in a pretty substantial proportion of those cells. I would say for some of the modifications, it's around 25% of all those cells. For others, it's getting up to 50 or 75% of those cells. So that can be really effective. You put those cells back into the bone marrow. They have advantages over the cells that are still carrying the sickle cell mutation. And so the patient is likely to do very well. So I don't know whether you may have seen a 60 Minutes production several months ago that was about a gene therapy for sickle cell disease. And they were doing exactly that sort of procedure, taking out bone marrow cells, treating them in the laboratory, and then putting them back into the patient. That particular procedure was being done with a gene therapy where this didn't involve CRISPR or zinc-finger nucleases, but just the use of a virus that carried the normal gene for the beta globin protein. So putting the normal gene into these stem cells that already had the sickle cell mutation is a way to moderating the effect of the sickle cell mutation itself. And the very, very early trials with this gene therapy are looking pretty promising too. So this was a 60 Minutes production on CBS, and you and your listeners might want to go back and look at that and just see the kind of things that are being done. So that particular procedure is not a correction; it's a gene addition, but the gene corrections are going to be done with exactly the same sorts of procedures.
So you can do a minority population of bone marrow cells, alter them, and they'll slowly alter the rest of the cells in the marrow, for instance. You can do at 20-25%, or you have to go higher.
So if you're at 25%, 50%, that's enough to overcome the effects of the sickle cells that will still exist in the bone marrow. And that's partly because the cells that have the correction are just healthier in the circulation. They last longer, and their presence in the circulation will sort of overcome the effects of the cells that still carry the sickle mutation. It's also true that if you go into the cells that carry the sickle mutation in both copies of the beta globin gene and you just correct one of them, that's a full correction. People who are naturally heterozygous, one sickle mutation, one wild type gene, they're fine as far as their red blood cells are concerned. So a partial correction can still be extremely effective.
And going back to the mechanism of action, so one mechanism is to cut the DNA at a certain point and then the cells naturally will fix it in the right way. Is that one mechanism?
Well, we have to provide the piece of DNA that's going to provide a template for the fix. So it's one of the other issues, and one of the things that probably limits the efficiency is we're providing both the reagents that are doing the cut and also a piece of template DNA that is going to be the sort of Band-Aid that fixes the break and provides the correction.
Okay, but you said doing the repair itself, that's difficult. Like, why is that difficult? Why is it easier to just include the, snip and then include the fragment and just have it resident there and then the body do it itself? Why is it hard to do the repair on our own?
Yeah, a lot of cells want to do a different type of repair that doesn't include copying sequences off the template DNA that we provide. And what that other mechanism of repair is basically just jamming the ends back together. And sometimes that is inaccurate, so it makes a mutation at the target site that's been cut, and that doesn't provide a correction. It's just it would be okay if at the same time, if we mutated one gene by this jamming back together mechanism, but got a correction on the other copy of the gene, the other allele. But it's this competition with this other mechanism of repair that limits the efficiency.
I see. I guess like that would be probably analogous to the effects of radiation on your body. It would cause breaks in the DNA, and then your body would, jam stuff back together, as you said, as one mechanism, and maybe that would increase the incidence of mutations that you have in DNA damage.
Right. And the good thing about using CRISPR is that the break is made at a single location. With radiation, you're just getting breaks all over the place. You have no way to control where in genomic DNA the damage is occurring. But with CRISPR, we make a break at the target site that we're aiming at. There may be breaks at other sites in the genome. That's the concern we were talking about earlier. But there is this additional concern that at the site we're targeting, when we make a break, we don't always get the outcome we want. We sometimes get a mutation there rather than the correction. And at this point in many cells, mutagenesis at the target site can be more efficient than correction. So people have done a lot in improving the efficiency of correction. So when I was working on a sickle cell project 4 or 5 years ago, we were getting correction at the rate of 2 or 3% in the cells that really would provide a therapy. Now that correction is up at 25, 30. I've even heard people say 40% in the best case. So we went from 2%, which would not have been therapeutic, up into a range that is therapeutic. And so the group that is continuing to work on that project is talking now about doing, gearing up for the initial clinical trials.
Yeah. Okay. So what are zinc-finger nucleases? I only heard about them. What do they do and how do they work versus CRISPR?
They work very similarly to CRISPR. The nuclease makes a break at a target site and then relies on cellular repair mechanisms to get mutagenesis or correction. Zinc-finger nucleases were the first of the platforms that you could really direct to essentially arbitrarily chosen sites in the genome. And they're still being used. There's a company in California called Sangamo Therapeutics. Their platform is the zinc-finger nucleases. They're using them in therapies, both ex vivo and in vivo, for particular disease conditions. So the zinc-finger nucleases are effective. They're just harder to use, harder to design for new targets. And so many people have switched to CRISPR because it's just a heck of a lot easier to manipulate and get it to make the on-target breaks that you're interested in.
Who discovered CRISPR? I heard it came from bacteria who use CRISPR.
That is true. So discovering CRISPR, the first clues that there was something out there that was kind of unusual in bacteria and ultimately led to the discoveries that we're making use of today. That discovery of a piece of the CRISPR stuff in bacteria, that was published in 1987 by a group in Japan, but they had no idea what it was all about. And it took more than 2 decades to get to the point where we really understood what CRISPR was doing in bacteria, how it operates, and how it could be adapted for use in cells of essentially any organism. And the people who are rightly given credit for discovering the essential pieces of the CRISPR system are Emmanuel Charpentier, who's now in Germany but was in Sweden when this work was done, and Jennifer Doudna, who's at the University of California in Berkeley. Mm-hmm. They published a paper in the summer of 2012 that showed how the system works and how you could adapt it to be used as a genome editing reagent.
Wow. Okay.
And they realized at the time that the work that had been done on zinc-finger nucleases many years before, and there was another technology that came between the zinc-finger nucleases and CRISPR that was called TALENs. But what was done with zinc-finger nucleases and TALENs made it absolutely clear to people how you could use CRISPR. So as soon as they saw that these are the few things you need to get CRISPR to make a targeted double-strand break, it could be used just like the ZFNs and TALENs. And so immediately, essentially, a number of different groups showed that you could use CRISPR in human cells, in mouse cells, in a bunch of other different organisms. And because of the simplicity of the CRISPR system, how easy it is to use, it just took off like gangbusters.
Yeah. Well, I read now they're able to start modifying RNA. I had just read this literally a couple days ago.
Yeah. And so one of the amazing things about CRISPR systems is that they're extremely widely distributed in bacteria and in archaeal organisms. And there are people that are working on just discovering as many of these as possible. And as more and more are discovered, it's being learned that although they are related systems, the way they function can be fairly different. And some are targeted at RNA, some are targeted at DNA. Some of them, when they target RNA and DNA, just make a very specific break. Some of them are activated to essentially cause the death of the cells where they're activated, so that a virus that has invaded the cells won't be passed on to other bacteria. So it's really an amazing system. And as people learn more about how they work in their home organisms, they're being adapted to work in experimental organisms and in people.
Oh. So what's your current research at this moment? What's involved?
Well, I closed my lab last year, so I'm not doing any laboratory research. At the moment, I'm continuing to work on some of the outreach and engagement efforts, the ethical and legal aspects of the genome editing technology, both in humans and in agricultural organisms. I'm quite interested in things that are being done in plants and animals as well as in humans.
In general, do you feel like we're playing with fire, that things aren't nearly well understood enough, that we're not going to have a lot of side consequences, or is that just an unnecessary part of the learning? I mean, what's your overall thought about genetically modified organisms and gene therapies?
Well, talking about gene therapy, I think we're slowly going at it. I use the we very broadly. I'm not engaged in it myself right now, but I think people are going at conditions that can be treated with pretty straightforward approaches, and they're going at it very slowly, very deliberately. So if there are going to be side effects that we don't anticipate, then we'll learn about it in the early stages of trying to develop these therapies. I'm optimistic about it. I think that we have enough control over the specificity of CRISPR and the zinc-finger nucleases that we can pretty well anticipate the adverse effects. And if it turns out that there are some we hadn't anticipated, there will be ways to get around those, avoid those. So I'm pretty optimistic about the early therapeutic applications. For broader therapeutic applications, there are going to be huge limitations based on delivery. How do you deliver the CRISPR reagents to exactly the cells you want to make modifications in? And I think that's going to be a real area of intense research. And until we have solutions to that challenge, it's still going to be sort of a niche therapy. In the agricultural applications, I think progress is going to be much more rapid, and the impact is going to be greater in the short term. There are already modified plants and animals that are out there. There are a few plants that are making it to market right now. There's a soybean that has an oil that's healthier than the standard soybean. The modifications that have been made are just to knock out some genes in the soy plant that are involved in the production of the oil. When those genes are knocked out, the oil that's produced is more like olive oil in its fat content, and so it's inherently healthier. Those soy plants are being grown in the field in the U.S. right now. There are mushrooms that have been modified so they don't turn brown when they're exposed to air. My understanding is that those are close to being in the market. And there are modifications like this that are being made in crop plants to just improve their characteristics. The other things that people are working on are perhaps knocking out the proteins in peanuts to which people are quite allergic. If you could make peanuts non-allergenic, that would be an improvement. People are working on making crops resistant to viruses that can wipe out these plants, particularly in the developing world. The developing world is where crops are much more insecure. An infestation with insect pests, an infestation with viruses, an extended drought can just wipe out food supply in areas of the developing world. One of the areas that people are working on quite hard is to try to address those susceptibilities in those crop plants and improve food security in those areas. And to the extent that that can be done, I think it's going to have a huge impact.
Yeah, I wonder if you could deliberately engineer in diversity to, let's say, rice in a given country. So you'd make very similar, types of rice plants, but they are different. And maybe that would help blunt the effects of, bugs and parasites and all kinds of stuff that would destroy the crop, just because the population is, more diverse than just one single type of rice plant, for instance. Is there any thoughts of doing something like that?
Yes, and one of the nice things about the genome editing technology is that you can make a modification that will improve the traits of the plant. You can make that in any cultivar. You don't have to do it in one standard type of rice, for example, and then try to distribute that rice all over the world. You can make a modification that might make the rice pest resistant in a rice that's already adapted to a particular environment. You can make that modification there. You can make that same modification in rice that's adapted to a different environment. So you can retain the diversity of rice crops that are adapted to different environments without having to introgress just one standard variety. And so it's a way to increase diversity rather than to move toward monoculture, which is something that people in agriculture are quite worried about.
And what kind of creature would you say is the best gene editor on the planet? Is it viruses? Is it bacteria? And for gene editing, again, we're using CRISPR, so it's bacteria-based. Viruses seem to be really good at it.
How would we deliver the genome editing? Is that what you're asking about?
Yeah, I guess, are there any other technologies similar to CRISPR and viral vectors that you see out there that, show promise but just aren't there yet?
Some characteristics of viruses are being used in delivery of the CRISPR reagents. So there are viruses that efficiently get into cells and then stay there for a long time. And that's in some cases what you want when you're delivering the CRISPR reagent. You want them to stay there for quite a while. That's particularly true of the version of CRISPR where you're modifying RNA. When you modify DNA, once you've done it, that's it. Those modifications will stay there as those cells divide and are maintained over long periods of time. But with RNA, there's new RNA being made all the time. RNA is being degraded and synthesized. And so if you're going to rely on modifying RNA, the ability to modify has to be there continuously. So people are using viruses that get into cells and stay there, that will continue to produce the CRISPR reagents over long periods of time. Viruses, as far as I know, natural viruses don't carry proteins that are good at targeted modification of cells. And so that's what we've borrowed from bacteria, or we've borrowed from capabilities of mammalian cells themselves to do these genome editing modifications. And we're using viruses to deliver those reagents, deliver the CRISPR reagents, deliver the zinc-finger nucleases, because viruses know how to get into cells.
Yeah, when I read a little bit about endogenous retroviruses, like it's amazing that viruses can integrate their own DNA into our DNA, and that could last for generations or forever. So I guess they hold promise to really cause significant alterations if they can be harnessed.
Yeah, the thing about viruses that integrate into cells, including the retroviruses that still integrate into our cells, these endogenous retroviruses are occasionally activated to make new copies of themselves. The problem with viruses that integrate into our genomes is they don't go where we want them, and we can't control where they go.
Right.
So they can make good vectors for some purposes, but what we'd rather do is what CRISPR does, which is to put things into the precise location where we want them.
Yeah, well, maybe we can harness them in some way where they can specifically do our bidding. So that's why I was just postulating maybe it's possible in the future.
People are working on that, and if we had a whiteboard that both you and I could look at, I could draw you some pictures. But viruses are being used in a lot of ways to support the CRISPR technology. But the key feature of CRISPR, this ability to target a unique location in a huge genome, is something that viruses don't do well on their own, but they can be an adjunct to help CRISPR.
Okay. Well, very good. Any last topics or questions that you want to cover that we haven't covered? Because I know time's running out. So
yeah. So I think CRISPR and the related technologies have a huge amount of promise for medicine, for agriculture, being used extremely widely in research. In the practical applications, I think one thing people need to keep in mind is that it's going to take some time for the benefits of the applications to really be manifested. It's not going to be tomorrow where you can check into the clinic and expect to get a CRISPR therapy. There's a lot of work to be done to make sure the therapies are safe and effective. And the way new therapies are tested in the US and most places around the world is to just go slowly, test a few patients at a time, make sure things are safe in the first instance, then go to a small number more patients and make sure there's some efficacy, sort of test out doses. And so it's going to take years for the clinical applications to really be available to people in the clinic. And so people shouldn't get too optimistic about when these things are going to be available. And if it turns out that there are some adverse effects that we haven't anticipated, that'll slow things down even more. So just be patient.
Okay. Well, very good, Dana. Well, I appreciate you taking the time. It's been a good interview, and thank you so much for being here.
Well, thank you, Richard. I appreciate the opportunity to talk with you, and I hope your listeners find it interesting.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.